Okay, this video is going to be about the difference between organic and inorganic compounds and also going to be about carbohydrates. So organic compounds tend to be complex and much more complex than inorganic compounds, and they all contain carbon and hydrogen. In order for something to be considered organic, it must have carbon and hydrogen, and they are always bonded together covalently. Many of them also have oxygen and nitrogen, and some of them have sulfur and phosphorus as well. Inorganic compounds usually don't have carbon in them. There are some exceptions, for example, carbon dioxide and calcium carbonate, but if you notice, there's no hydrogen atoms in either of those molecular formulas. So typically they have they always have carbon and hydrogen, and they're typically more complex than inorganic compounds. There are different types of organic compounds. We have carbohydrates, which have a 2-to-1 ratio of hydrogen to oxygen. That is almost always true. There are a few exceptions to that 2-to-1 ratio of hydrogen to oxygen. For example, the sugar that's in DNA, which we will get to later. But there's a reason why it's called a carbohydrate. It's basically carbon. And then the hydration or the hydrate is a reference to the hydrogen and oxygen in the same ratio that water is. And you know if something is dehydrated, it doesn't have enough water. And if you hydrate something, you add water. So monosaccharides are simple sugars. The prefix mono means one. And the simple sugars have a molecular formula of C6H12O6, which tells you the different types of elements and the ratios of those elements. So it is a 1 to 2-to-1 ratio between carbon, hydrogen, and oxygen. But there are many different arrangements of these atoms. Monosaccharides contain a large amount of energy in their bonds that can be released in the presence of oxygen to make a molecule called ATP. ATP stands for adenosine triphosphate, and that is considered the energy currency in cells. So we, in our bodies, break down glucose to make ATP, and then our cells use ATP to do work in our cells. And we are going to be spending a lot of time talking about ATP in future units. So right here is a picture of glucose. Here's a 3D model of glucose. Now, I would like it noted for the record that everywhere where you have these corners, there are carbons there, but they're not drawn. Biochemists know that there are carbons at those corners, and so we don't have to draw every atom. Glucose is a monosaccharide, galactose is a monosaccharide, and fructose is a monosaccharide. The difference between glucose and fructose is very obvious. The shape is different. Glucose's ring structure is a hexagon, whereas fructose is a pentagon. So that's pretty easy to tell the difference. The difference between glucose and galactose is this position of the OH and the H. The OH is called a hydroxyl group, and then that's a hydrogen atom. And if you see, they're flipped from one another. You will not be tested or quizzed on the difference between glucose and galactose. It would be very tough to remember. And if I'm honest, I have to look that up sometime. So you won't be tested on the difference between these two molecules. These, maybe. Okay, so each of these monosaccharides has the same molecular formula. They're all made out of 6 carbons, 12 hydrogens, and 6 oxygens, but they have a different structural formula. So molecules with the same numbers and types of atoms but different structures are called isomers. Be really careful because it's very common for students to put the word isotopes on tests and quizzes instead of isomers. Remember, isotopes are atoms of the same elements that have different masses because they have a different number of neutrons, not the same thing. So just be really careful. So again, glucose, galactose, and fructose are isomers of each other and have the same molecular formula but different structures. So how do we end up with complex carbohydrates? Well, dehydration synthesis is how that happens. So dehydration synthesis bonds sugar molecules together. So synthesis means to put together, and dehydration means removing water. So if we show you this diagram, we have 2 glucose molecules, and if the hydrogen comes off of one hydroxyl group and the OH comes off this molecule, the hydroxyl group, comes together, and you have 2 hydrogens and 1 oxygen, and you get water. So now you have a disaccharide, so you have maltose. So if you have glucose plus glucose, you get maltose. So a disaccharide, the prefix di means 2, saccharide tells you it's a carbohydrate, is 2 simple sugars joined together. So maltose is 2 glucose molecules. A sucrose is a glucose and a fructose. So you would have a hexagon and a pentagon when you can tell this is maltose because it has 2 hexagons. One thing that you'll start to notice as we go through more and more carbohydrates is that most of them end with "-ose" at the end. So their specific names always end in "-ose, but if we're talking about the general categories, whether you have a monosaccharide, a simple sugar, like one ring, they end in saccharide, and if you have 2, it's disaccharide. And those are more general terms, whereas maltose and sucrose are 2 different disaccharides. This is their very specific name. The category that they fall into is disaccharide, and then the larger category that they fall into would be carbohydrates. So a polysaccharide is a polymer of monosaccharides, often glucose. It's almost always glucose. My daughter remembers that by glue, like think glue sticking things together. So she came up with that little memory trick last year. Starch is a polysaccharide that's used for the storage of energy in plants. If you've ever seen a container of cornstarch, which is often used for baking, you know corn is a plant. So then you can think to yourself, ooh, cornstarch. Starch is how plants store their energy. Glycogen is used for the storage of energy in animals, and it's stored in our liver and muscle tissues, and it's more branched than starch. But typically, any diagram of starch or glycogen will say starch or glycogen because they are very, very close cousins of one another. The only difference is that glycogen is slightly more branched, and so in order to tell the difference, you'd have to look at 2 side-by-side diagrams, and you'd look for the one that has more branches, and you would know that that is glycogen. But they're very, very similar. One thing that you should notice, not only is that there are branches, but that if you look at the glucose molecules, they're all facing the same similar direction. If you look at the oxygens in the glucose ring, they're all facing the same direction. And that's going to be important to remember when I show you cellulose in contrast. So I want to show you a little video of dehydration synthesis. So water comes off, or hydrogen and OH come off. The bond is formed between the 2 monosaccharides. You have a water, and now you have a disaccharide. Now, once you get more of those, it is considered a polysaccharide. Now, notice that if you've got 5 monomers or 5 monosaccharides, you get only 4 water molecules because you only have to remove H's and OH between each molecule to get them to come together. So however many monosaccharides you have in your chain, you will always have 1 fewer water molecule. So 5, and then you have 4 water molecules. So that is a glucose molecule. So glucose is the monomer of this polymer. So here's a monomer, polymer. So I've been using the terms monomer and polymer, and now I really want to kind of focus in on what those terms mean. So a monomer is a general term for a building block of a polymer. And the polymer is a general term for a long repeating chain of any building block. So here is a general representation of a monomer. And then if we put a bunch of them together, we have a polymer. Now, this isn't actually in the notes, but if you have 2 of them, it's a dimer. So mono means 1. The prefix di means 2. So this would be a dimer. And then now you have your polymer. Now, monomer, dimer, and polymer are general terms that can really apply to all kinds of molecules that are organic. And then when we say monosaccharide, disaccharide, and polysaccharide, we know we're talking about monomers, dimers, and polymers of carbohydrates specifically. And then when we name them, like glucose, and then maltose, and then starch or glycogen, then we know specifically their exact structures. So again, monomers and polymers are general terms that apply to many biochemical molecules. So the third polysaccharide that I want to talk about is cellulose. Now, cellulose is important for many reasons. In the plant kingdom, it's important because it's the structural component of cell walls. It's what makes them crunchy, but also strong. So wood is really dead cells, but what's left is the cell walls that are made of cellulose. It can't be digested by us, and it's considered dietary fiber. If you look at this molecule, you'll notice a couple things that make it different from starch and glycogen. It doesn't have any branches. It's long, straight chains. Now, of course, cellulose is really much longer than this diagram would seem to imply. But notice it doesn't have any branches. And you should also notice that the oxygens, here it's in the top, now here it's at the bottom, here it's at the top, here it's at the bottom. So they're kind of like upside down, alternating, right side up, upside down, right side up, upside down, right side up. Whereas in the starch, all of the oxygens were on the same side. And this bonding creates different bond angles here that our body has a really hard time breaking. Actually, we can't break these down. So when we eat dietary fiber, we can't break the glucose down into individual units, and it doesn't get absorbed into our cells, and we can't get energy from it. So that would be bad if we were if we didn't have enough energy. But with the modern American diet, where we actually, don't move around as much as our ancestors did, and we don't have as much exercise, and we actually have an abundance of high-calorie foods, dietary fiber is actually very good for us for many reasons. First of all, dietary fiber makes us feel full and takes a very long time, sits in our stomach for a long time. So it makes us feel full for a long time. It keeps us from overeating. Also, when you eat sugar with dietary fiber, it absorbs into your bloodstream more slowly and keeps our blood sugar stable, which helps us maintain a healthy body weight. But it also prevents colon cancer. And in addition, most foods that are high in dietary fiber are also very, very high in other nutrients and vitamins and minerals. You find dietary fiber in things like fruits and vegetables. So when you're eating an apple, the dietary fiber in the apple is good for you, but there's also lots of other micronutrients that are very, very healthy. So if you find a high fiber food, it's probably got other things in addition to the fiber that are very good for you. So hydrolysis is the opposite of dehydration synthesis. And we're going to see hydrolysis and dehydration synthesis throughout this unit many times, no matter really what class of organic compounds that we're talking about. But hydrolysis is when water is used to break down polysaccharides into disaccharides and monosaccharides. It is the reverse of the process you've seen before. So here, if you look at this is a disaccharide. I know that happens to be sucrose because that's a glucose and that's a fructose. I kind of think it looks like an ant. And remember, ants love sucrose. Sucrose is table sugar. So water will go in and break that bond, and we'll end up with two monosaccharides, a glucose and a fructose.